Our collaborative acute COVID study in 2020 identified key immune signatures that corresponded to COVID-19 disease severity. These were evident upon admission before patients declined into severe disease and critical care. One of these immune changes was a distinct alteration in monocyte function, and this was most striking in patients destined for intensive care. Since this time, we've followed up patients that were hospitalised with COVID-19 for up to 9 months post-discharge to determine how long this monocyte function is dysregulated for, and how this monocyte dysfunction may correspond to long COVID symptoms, which continue to affect a substantial proportion of people following COVID-19. This work has been a huge collaborative effort involving several other lab groups within the university across divisions, including CIRCO members who are the Lydia Becker immunologists, clinician scientists, research nurses and bioinformaticians. We have follow-up samples taken from 4 different hospitals across Manchester. We have found distinct changes in monocytes, in particular in migration patterns that allow them to infiltrate peripheral tissues. These are abnormal in convalescence in COVID-19, and these abnormalities correspond to distinct symptoms of long COVID. In particular, we have found 2 distinct immune signatures that correspond to the 2 most predominant symptoms of long COVID, shortness of breath and fatigue. These different immune signatures will help us develop more targeted therapeutic approaches for patients who continue to suffer with these specific long COVID symptoms.